Okay, Professor Sharp, thank you for being here at the University of Virginia and your lecture and the meeting with the students, the faculty, and we're delighted that this visit actually happened after 2 years of planning. And the focus of this conversation is to really, go beyond and learn from your lived experiences and how you made certain transitions, decisions that led you from the college all the way to your accomplishments of the day. So I'm just going to ask a few questions and see if we can navigate through this with a period of time. Yeah. Phillip,
so we were getting echoes off this screen. Are we not echoing now? Can you hear?
Yeah.
Oh, okay, fine.
Okay, great. To kick this off, let's go back to the early years and talk to us about what motivated you, your background and what motivated you to go to college and who the influencers were starting at that point.
Well, you have to understand where I came from. And I grew up on a small farm in northern Kentucky, about 40 to 50 miles out of Cincinnati, on Route 27. The town nearest by, about 7 miles away, was called Falmouth. And my mother's family lived in that part of the country for about 110 years, back in a small community 10 miles out of Falmouth called McKinney'sburg. And my father had lived several generations in that area. My mother had an 8th grade education. So when this was before she graduated from the 8th grade, before there was school bus service that would allow her to be taken to a high school nearby, not nearby, quite distant. So she ended her education. My father went through high school in the local town. And so I grew up for the first 6 months, 6 years. My father was a tenant farmer on my mother's family farm. And then we bought a farm separated but in the same area. And I worked on the farm raising tobacco, though I spent my whole life in a cancer center. And we had animals. We milked some cows and sold cream and raised animals and corn. And I went to the local high school and public school. When I was a sophomore, it was consolidated. My freshman year, I had 16 students in the class. We consolidated to 60. So it was, in high school, well, let's put it this way. I really became oriented in science when I was in about 10th to 11th grade. I enjoyed thinking about the world in a quantitative way, talking about numbers, sizes, all sorts of aspects. I was one of the strongest in a small group in mathematics. And when I got to high school, I was the strongest student in chemistry and physics. I had a math teacher, Mrs. Haber, who took special interest in me. And in my junior and senior year, a young high school teacher, Mr. Kellums, came in, and he taught chemistry and physics as well as higher math. And I spent a whole day with him. And he encouraged me, but my parents, from the time I was 6, encouraged me to go to college. So I'll give you a story that'll illustrate this. I had been saving money in what was a rooster bank as I was a kid. So I would get a nickel and I'd save it, and it was to pay for college. That's what my parents encouraged me to do. So when I was 6, my grandfather had gone to Falmouth, the local town, and brought back a little calf, female calf, which we named Blackie. And I paid $16.75 for that calf. From my piggy bank. It broke the piggy bank. There was nothing left in that piggy bank. But from that time on, they milked the cow and sold the milk, and I got the calves, and I would sell the calves and save money to go to college. And they gave me a quarter of an acre of tobacco to grow. And I grew that, and I helped them with the rest of it, and I saved that money. So by the time I got into high school, I was oriented to go to college. I really didn't know much about, though I'd been on campuses because I played basketball in high school, and I made the varsity team, and it was all, this is Kentucky, and there's nothing bigger than Kentucky than a basketball team. And so I had lots of friends that way, but I applied to one college, Union College in Barboursville, Kentucky. It's in the mountains. And luckily they took me, or there would have been a different history. My counseling in high school, the principal called me in. 2 times he called me in my high school. That's good, because you usually got called for bad things. He called me in and asked me if what I was going to do, and I said I applied to Union College, and he said that's fine. And that was my counseling. The second time he called me in, and he said the Chamber of Commerce has asked me to ask you if they would help pay for your medical training. Would you come back to the county to be a doctor? And I said I was much too young to make that decision. So I said no. And so I applied to Union, and they did admit me. And it's a small Methodist church-related college in the Kentucky side of the Appalachian. A beautiful place, small college. I got a lot of attention, a lot of encouragement, but not a great education in chemistry and math, which is what I majored in. And I was one of the strongest students there. So I'll continue this story. We had to borrow money. By the time I was in college, as I graduated, when I was 20 years old, I got married. So it was Ann and I, and we were watching numbers, so I graduated mid-year in 3.5 years from college. I applied. I was fascinated by the idea that I could continue to learn as a graduate student, and I wouldn't have to pay tuition. And so it seemed like just a wonderful environment to be in. So I applied to several Midwestern schools. I wasn't confident enough in myself to go to Stanford or the East Coast. I didn't think I would be accepted anyhow. So the University of Illinois accepted me, and in their mid-year in January. And I go to Illinois in chemistry, which is a great department, one of the first top 5 or 6 in the country by ranking. And like most chemistry departments do, they give you an entry exam. How proficient are you in the 4 areas of chemistry, and I failed 3 of the 4. I just had never seen the material. I never had a chance to learn it. And they did exactly what they should have done. The chair of the department said, Phil, go take our senior classes. And so I took their class in inorganic chemistry, analytical chemistry, and physical chemistry, all in one semester, and just blew it away. I was really motivated. And so I entered the graduate program and started working on biophysical chemistry. That's what I was interested in. And in 3 years, I got my degree.
Oh, that is, I mean, that's remarkable. 3.5 years undergraduate. I mean, is there a reason why you went through, or did you 3.5 years undergraduate, or because you're able to handle the courses and you're done, or was there other reason, financial? I don't know.
Well, everything was. This is the Vietnam War. You know, this is when draft was a very important thing. It was just ending as I was getting my degree. Resources was always an issue, but I had mastered the material that I was working on, the hydrodynamics of DNA chains, long, stiff polymers, which is what I was working on. And I had decided at that stage, by simply reading books about molecular biology, and particularly chromosome biology, which the Cold Spring Harbor Symposium back in the day, one of the most important meetings was always the Cold Spring Harbor Symposium. It was about a particular topic. There was a big book produced, review articles about the field, and I had picked up that review about chromosomes. And I decided from the biophysics perspective, I could do a lot of good science in chromosome biology. And I wanted to learn molecular biology. So I made that as a decision, and didn't see a reason to not move on and learn the next thing I was interested in learning.
So that really is a decision based on your interest and inclination to move into life sciences at this point, after a PhD, from a strong chemistry, polymer chemistry, to life sciences at Caltech, right? That's your first postdoc.
I was accepted by Norman Davison at Caltech. Norman was a shock tube chemist, and then a inorganic coordination chemist. He got in the National Academy when he was 40. He was one of the, I just loved his papers. I would read his papers, and I just loved the way he went at a problem, analyzing it from chemical principles, thermodynamics, other chemical principles, and then solving problems in molecular biology from that perspective. And just before I went there, Ron Davis, who is from Midwest, he's from Southern Illinois, had been a graduate student with him and had developed the first genome map using electron microscopy, mapping deletions on a phage genome. And that's a paper in the late 60s, first mapping of a deletion on a chromosome. And that I thought was a whole new technology that allowed us to move from not seeing chromosomes and genes as chemical, physical entities, to being able to do that with DNA. So Norman's lab then became a very attractive lab. So I moved to Caltech.
Yeah, and you obviously worked with lots of people in the lab that enabled you to develop your skills and contribute too. Can you describe the environment that you had where you did all the good science you did? What's that environment like?
When I went to I started at Caltech, so Norman was in his more mature years. Let's say he was about 60 when I went there, maybe a little older. And he had a research lab of 20 people. And most of them were engaged in some aspect of biological science, mostly oriented towards genes and chromosomes or DNA renaturation and various things of that nature. Now, his life path was interesting because he was as I said, shock tube physical chemist and then an inorganic chemist. And he got throat cancer when he was in his 50s or early 50s. Went to Chicago, took radiation, was cured, but decided at that stage to switch his research interest to biology and started bringing physical chemistry to DNA. And so he had a chemical environment, but a strong physical chemistry approach to life science, but in a department or in a location, because he was located in an area with Jerome Vinograd, who had discovered superhelical DNA. And here was a group of 50. And I was in Norman's lab, but shortly after I was there, maybe 4 or 5 months, I got to know a lot of people in Jerry Vinograd's lab. And so I went to Vinograd and said, I'd like to attend your group meetings too. And then asked Norman if I could attend both of them, and both of them were very pleased to do that. So I had an environment of 50 people. I started doing experiments mapping genes on chromosomes in E. coli, the total E. coli chromosome. Made major progress in doing so. I mapped lambda integration, I mapped LAC and many other genes while I was there. And I wrote for reagents to Japanese new PhD postdoc for bacterial plasmids, F primes, that he had studied. He sent the reagents and he sent a letter saying, I'd like to come as a postdoc. So he joined Vinograd's group. A new graduate student joined the research program. So there was 3 of us all working together on bacterial plasmids. I collaborated with Stan Cohen at Stanford, the Cohen and Boyer Cohen. And we mapped drug resistance markers on plasmids that move plasmids around. And I like interacting with people. I just enjoy sharing ideas and helping each other out. And so things went very well.
Well, that's great. I mean, at the end, I mean, there is a theme to it. I mean, mapping genes on DNA using electron microscopy. That all the way, the technique.
Made a big advantage.
The big one, right?
Yeah.
So you've got that. So did you? Is that technique something that you improved further as you did
Yeah. I learned how to do. I really learned how to analyze very complex systems, very big chromosomes with that technology. And so I was there for 2 years, in 1971. 1970 wasn't a good time to go on the job market. We were in a depression, recession at that year. There were very few people were uncertain about NIH and whether NIH would continue to grow in funding. I wasn't interested in going to a university where I couldn't do research. All right. So I applied to a number of jobs, got interviewed at Stanford in chemistry. And the good news is they didn't offer me a job. Because if I'd have gone there, the pressure would have been to continue what I was doing as a postdoc. But I'd already made the decision that I was going to move from bacterial systems into mammalian cell systems because I knew I could do good biophysical approaches to chromosomes in mammalian systems, particularly using viruses. And so SB40 was purifiable with superhelical DNA. You could do it. I did some microscopy with Jerry Vinograd. We published an interesting paper together. So I decided I really wanted to make the transition to mammalian systems, but I had really never done anything. I never even cultured mammalian cells. So after a few job interviews, I wrote to 3 people, Howard Tinman, Dave Baltimore, and Jim Watson, and asked said I wanted to do a postdoc for a couple of years to learn how to work with viruses in mammalian systems. I got the exact response, if you know those characters, which I learned later. Howard Tinman wrote, if you want to come and talk, we can talk about it. Howard was a very particular person, a wonderful scientist, a deep thinker, but a very particular person. David responded, very interesting. I'd be interested, but I'm going to be full for the next 2 years. So if you could wait a little while, we can do something. And Jim Watson, who had just taken the directorship of the Cold Spring Harbor lab, called and said, I'm going off to Europe in 2 weeks and I'm going to recruit a postdoc. If you want the job, call tomorrow and tell me if you want it. So I went to talk to my faculty, actually Bob Sensheimer and others in biology, and asked them about what was going on at Cold Spring Harbor and various other things. Jerry Vinograd told me about Joe Sandberg, who was a young person had just recruited there, who was very good. And Sensheimer said, anything Jim Watson's doing, Jim Watson's going to do well. So I called back the next day and said, I'm coming. And we arrived in June in 71, and I worked there for 3 years. Yeah, at Cold Spring Harbor, where I learned virology and cell biology.
Yeah, this is the Watson of the Watson and Crick.
Yes. Yeah.
Just to be.
Gen mentoring is a very interesting process.
Tell us about it.
He said, go in the lab there and find something to do. So I soon found Joe Sandberg as someone who was a very good trained, experienced virologist. He was actually trained in myxovirus in Australia, and then went back to Sydney Brenner in Cambridge, and was there 2 years, and then went with Renato De Becco at Salk. And he was a deep virologist, but he really benefited from collaboration from somebody who was more biophysical as a chemist, particularly because what we could do in breaking down these viruses was to actually use RNA DNA hybridization. And I understood RNA DNA hybridization and how to separate strands of DNA and how to map percent. And so we started collaborating and the papers started flying. And it was, and then 1 year after I was there, they made me a staff member at Cold Spring Harbor. And then Jim would show up, at midnight or something in the morning, and late in the evening, and he'd stop in your door, and he'd look at you and say, tell you a story about something from Harvard, and then ask you what was interesting. And you tell him, hopefully you tell him something that's working. and he would, And then, at a certain stage, he'd say, you ought to write that up. And so I and the collaborators would write it up, and then he would say, you ought to give that to John Hershey to read. Hershey was an incredible editor of papers. And so I would leave it in Hershey's box. And Hershey would, about 3 days later, give me a draft back, and it was just red. What does this mean? DNA is not random. This other thing. And then you learn how to be precise in your language and precise in your thought. And that was very good training.
Yeah, you spent 3 years there, right?
I spent 3 years.
3 years. What would you say? I mean, when you do a stint, I always like to say, "That's my legacy. This is what I accomplished." Yeah. What would that be in your technical terms?
Well, we did a number of things. We were among the first to identify the regions of SP40 that was the early regions that had the oncogene activity and the late genes. We identified SP40 integrated in cells, in tumor cells, and we identified the early regions of SP40 as the only DNA sequence you found in tumor cells. We started collaborating. I wanted to work on a larger genome than SP40, so I was following virology, and I got interested in adenovirus because it's a longer genome. And Ulf Peterson, who was a postdoc from Leonard Philipson's lab in Sweden, came about a year after I was there, and we occupied an office together. And Ulf knew how to grow adenovirus and the molecular biology of adenovirus. And he was interested in adenovirus DNA replication. I was interested in its transcription and gene expression. So we started collaborating, and at that stage, Dan Nathans and Ham Smith discovered restriction enzymes, and then Boyer on the West Coast. So here we had enzymes that could break DNA apart. So I had been using ethidium bromide to stain DNA in SB40 preparations, and I knew how to use gels to resolve DNA. So I said, if you're going to isolate restriction enzymes, you could take the digested DNA, run it on an agarose gel, stain it with ethidium, and you could see the cleavage pattern. So this could be a big advantage. So I developed that technique. It's one of my most highly referenced. And started purifying restriction enzymes, and then fragmenting the adenovirus genome, and then mapping the adenovirus genome. And we identified the E1A regions, all the early regions, late regions, and transcription map while we were at Cold Spring Harbor. But after 3 years at Cold Spring Harbor, I really wanted to go back to an academic environment. I wanted to be next to chemists and biologists, and I wanted to be in a bigger environment. So I applied to a number of places, and I knew MIT was starting a cancer center, and Dave Baltimore was going to be one of the scientific leaders. Salvador Luria was going to be the director. And I was getting offers from other people, and some of them with tenure, some of them without. And I was waiting for a call. and finally it came, and I got a call from Luria saying, "We'd like you to come up and interview." So a couple of weeks later, I flew up and gave a talk, and about a week later, he called and said, "We want to offer you a job." And I flew up, the worst negotiation in the world. Here I asked, "Am I going to be on the same floor as Dave Baltimore?" "Yes." "All right. Do I get enough lab bay as a standard lab bay?" "I get 2 rooms, enough for 8 people." "Yes." "And you're offering me for a setup, and it was a small setup. I said, fine, I'll take it. Because if I had that environment, I could take care of everything else. I just wanted to be in that environment. I knew I would be able to attract really good people in that environment. I knew I would be able to get support and open with others around me. And at this stage, it was a new center, and there was going to be 11 young faculty recruited. Mm-hmm. And I very much wanted to be in a place where young faculty, at the end of the day, they're sort of too tired to go in the lab and just hang around in the hall and talk. Just talk about science, talk about what's new, talk about what they're fascinated by. Mm-hmm. And it worked. Nancy Hopkins shared an office with me. Dave Hausman was down the hall. Bob Weinberg was down this hall. Dave Baltimore was at the end of the hall. Richard Hines in cell biology was downstairs. And it was just a fabulous environment to do science.
Right. What you're describing is just excellence, amazing people doing their best.
Yeah.
And you want to be thrown into that environment. That's what you want.
The legend we were on the 5th floor of the cancer center. It was a new cancer center, and the legend was the lights never went out on the 5th floor. It was 24 hours a day. There was someone in the labs doing work. And the reason for that is we were so densely packed that if you wanted to get to the instruments, you worked different times. And so, I mean, it was a very, very stimulating environment to be in.
Yeah, I mean, that is just dwelling on that environment, that is the sort of environment institutions who want to build and do world-class science is trying to replicate and try to do, having a critical mass of diverse disciplines, experts to be co-located. I mean, you are big on proximity, right? I mean, that's an important element of this. Yeah. So speak to about the proximity of having people close by.
Well, I mean, okay, so I was at I had come from Cold Spring Harbor, which is a research institute, and everybody is, in one building or two buildings, and I organized the first, multi-group seminar there, ran the seminar series for a couple of years. So after I got to MIT, I had a postdoc, Jane Flint, who moved with me to MIT from Cold Spring Harbor. I hired an assistant. So there was 3 in the lab. I was going bananas talking to 3 people. So I went to Dave Baltimore and said, I would like to attend your group meeting because I just need more people to talk, and I don't know all the things you're doing, and I'd like to learn. So David said, fine. And then Bob Weinberg was already part of that group. So we started a weekly 1-hour group meeting every noon on Wednesday. Postdocs and graduate students rotated and talked. Because my lab was so new and I was doing research at the bench myself, I talked in that series. Now, fast forward 45 years, we're still having that group meeting.
Wow, that is remarkable.
Dave Baltimore's gone. He went off to be, president of Rockefeller and then Caltech. But Bob Weinberg, Dave Hausman, Rudy Yanish, Michael Yaffe are all part of that group. Jackie Lees, Stephanie Springer. So all our postdocs and grad students get up and speak to about 100 people in a room like this. It's wonderful training for them because they got to get up in a half hour, tell you the question they're researching, how they're doing it, what results they have. And it's a knowledgeable audience, but it's a a diverse enough audience, they have to give some background. And by the time they rotate through almost every year. So by the time they've been there 3 or 4 years, they are very comfortable getting up and just spieling what they're doing. It's a wonderful training environment. And I have seen magic come out of that meeting where somebody has been struggling for years to do something and somebody raised their hand and said, "Do you know?" And literally within 2 weeks, they've solved their problem. So it is a very important process. And every time something new happens in the broader scientific community, somebody in that room knows. So they will bring it to that room and talk about this new technique, that new concept, that new idea. And it brings the whole community up together. So it's really a very effective way. And I've had lots of postdocs and grad students leave my lab, but postdocs go off and establish their labs and develop a similar environment. That has been very useful for them.
I mean, that kind of points to speaking to a broader audience and expecting that what you don't understand, maybe somebody else has a perspective that enlightens you.
Yeah. Makes a connection, and will come up and tell you about it, or tell you about a reference, or send you a paper, or all sorts of things of that nature happens.
And Phil, going forward, your critical experiment has a story as to why you did the critical experiment that led to the Nobel Prize. Take us back to the experiment you're doing and the questions that you asked that led to this the beautiful chart with these loops that I see. I couldn't get over that picture ever.
So, as I described, when I moved from Illinois to Caltech, I started thinking about molecular biology from a chromosome perspective. And because that's what my training in biophysical chemistry had, it's where the strength was. And I became interested in a phenomena that was called heterogeneous nuclear RNA. Now, this was before your time. And Jim Darnell and a number of scientists had been interested in the synthesis of RNA in mammalian cells. And they were doing pulse labeling and trying to purify messenger RNA. And when they did that, the messenger RNA sequences, which they could map by hybridization at that stage, was in much longer RNA in the nucleus than it was in the cytoplasm. So, and it was, but you couldn't do a precursor product relationship because you can't flush the pool in the nucleus of a mammalian cell with cold isotope or cold radionucleotides fast enough. In 20, 30 minutes it took to equilibrate the pool. So the precursor product relationship wasn't established. They didn't understand it. And then there was the discovery by Mary Edmonds, who really didn't get enough credit for this, that message had a poly-A tail on the 3' end. And soon, Darnell showed poly-A tail on the nuclear RNA by just doing oligo-T purification. And then there was the discovery of a cap. And lo and behold, it was on the 5' end of message. And you look at the large nuclear RNA, it was on the 5' end of the large nuclear RNA. So, big puzzle. It was 5 times larger, and it had both modifications on both ends. We couldn't figure out whether it was conserved or not. But when, adenovirus is about 35,000 base pairs in length. And pulse labeling showed there was a massive long RNA produced in adenovirus. And it was produced late in infection where there was lots of it, right? So here I had a big genome I could do microscopy on. And I knew there was long RNA. And I And I knew it was capped and polyadenylated. And I said, I can figure this out. So we started working on using microscopy to map that nuclear RNA and the cytoplasmic RNA. And that was a paper that appeared in PNAS with Sue Burgett, postdoc in my lab, Claire Moore, who was at that stage a technician who came back to get a PhD in my lab, and me. And we found that there was a little tail at the end of the message, and it was very reproducible. Length was reproducible. It couldn't have been a random occurrence. And then we tried that foldback molecule, which told us that 3 pieces were spliced together to the body of the message. And because I knew that the hnRNP for cellular genes had a similar long structure and smaller structure, I wrote the paper from the perspective that this is how mammalian genes is expressed, and coined the term RNA splicing in the title of it, just so they wouldn't miss it. And so it was, I knew it was a very large impact paper, and that it would be. So I went to Cold Spring Harbor that summer to the symposium, made a presentation there. And by the time summer was over, there were 5 labs who had confirmed this. Most of those labs already had data in their notebooks they couldn't understand. And that just told them what the answer was. And they then did a few more experiments and said, this works for ovalbumin, chambone, this is globin, 2 or 3 labs for globin. It was a very exciting time.
That teaches an important lesson that many people did not understand, they just stopped there.
Yep.
And you pursued, this is not understandable, made hypothesis and just went ahead and verified it and got to the answer, right?
I mean, I was interested in that question. I was thinking about what is the fundamental process that is involved in the expression of genes, because I knew I needed to know that answer before I worried about regulation, because regulation works somewhere along the gene. It could be at the initiation process or the elongation, termination. All of those could be regulated. But if you didn't understand the molecular mechanisms and where that occurred, you weren't going to make any progress. And at that stage, we didn't know where a promoter was on any piece of DNA in mammalian cells. So once we knew splicing, the cap was at the promoter. Okay, cut the promoter, start doing biochemistry. That's what I did. And you needed a splicing mechanism. So I developed an in vitro splicing system so I could begin to analyze the components in the splicing system. And that took the next 20 years. And it was really exciting. Lots of things happened.
Yeah, and when you had the discovery and you see that it has changed biology and in fact, majority of the eukaryotic cells work like this. Yeah. It's not an exception, it is the rule. I mean, that's very powerful that you got to that point. Did that, as you know, Nobel Prize winning work, and by the time you get Nobel Prize for 20 years apart or 23 years apart, there's a number like that. Did you have a sense that did you have like an internal feeling that this is one of those that could lead you there, or you just kept going?
Well, I kept going because that's what I wanted to do.
Yeah.
And that was the interesting thing. But let me explain that process. So you make it, and it could be any number of things. David Julius, a contemporary Nobel just got announced. So you make a discovery. And the first thing if people think it is really an important discovery, you'll get nominated for some prizes. The Eli Lilly Prize, the National Academy Molecular Biology Prize. And when you get those prizes, you look at who has gotten it before, and there's a long list, and you can say, well, 1 in 4 of the people on that list got a Nobel Prize. And You know, if you go to Alaska, it's higher than that. You go to the Gardner, and it's higher than that. So you have to, I mean, it's not imagination whether you've been mentioned in this, because you've already been told people are thinking about the importance of your work by nominating you and supporting you for those prizes. So in this particular case, though, I and Tom Cech, now Tom Cech is a physical chemist, much like I came out of the background I did, but he came to MIT with Mary Lou Purdue and then went to Colorado and switched his research interest to splicing and found an intron in a ribosomal gene in tetrahymena. And he was interested in the splicing process, so he started purifying the RNA. And as he purified the RNA, he kept each step putting it back into a system that if you incubated the RNA, he knew in crude extract it would splice. And he kept purifying it and purifying it. And it kept splicing and splicing. And at the end of the day, there was no protein there, and he added it to the buffer and it spliced. And then he made the RNA synthetically and showed that it was self-splicing. So a dramatic discovery of catalytic RNA, which would carry out the two reactions in splicing, transesterification at the 5' end and 3' end. So we got the Lasker Prize together, and we got several prizes together. And then in '89, 4 years before I got the prize, he got the prize in chemistry for catalytic RNA. And so I thought, we all do good science. There's excellent science in the world, much more than can be, awarded any prize. So I assumed that was the recognition of the field, and the committees in Sweden would go on in different ways and do different things. So I stopped worrying about getting telephone calls at 6 in the morning on Nobel morning. And 4 years later, at 6 in the morning, I get a call. And I was sound asleep when it came. And it was a holiday in Boston. It's Indigenous People Day, but at the stage it was called Columbus Day. And so I stumbled over to the phone, and one of a colleague in Sweden that I knew, said that I had just been selected for the Nobel Prize and announced, and I said, could you repeat that? And he repeated it. And I said, thank you, and I'm very honored. And of course, my emotions is like this. And the next thing he asked me is, do you know the telephone number of Richard Roberts? Because Richard Roberts and I received the prize together. The Cold Spring Harbor Lab had done similar work in the same time period, and Richard was the leader of that work. And Richard had just moved to New England Biolabs, and I probably could have found over another half hour searching through everything his telephone number, but I said, no way.
You're not in a state of mind to look at a phone.
State of mind, I was not going to do that. My wife is excited, the kids are running around. So, no. So they found the number somehow. But it was quite a celebratory day.
Oh, that's fantastic. That's of course it's so special to you, that you got the call, and of course now I watched your lecture, Nobel lecture, and so on. Just amazing. Now a couple of questions. Maybe the next question is the timing of Biogen. This discovery, 1977. And you're at the pinnacle of this work, if you want to kind of interpret it that way. And you took the time to start a company and moving research from bench to bedside, so to speak. What was the motivation for doing that at that point when you were just way too busy just doing the science? You could have been continuing on. What motivated you to take that bandwidth and apply it for starting a company?
Well, the story there is in the spring of, it was late spring '77. I got a call from a man named Ray Schaefer. It was a beautiful day in the spring. My office at MIT was a southern-looking window, and I would go into the office, and if I wasn't in a lab, I'd be sitting in the office and I'd be reading, and the sun would come over my back. I was warm, and I was reading science, and it's just a wonderful place to be by yourself and learn. And I get this telephone call, and he calls me and says, I'm a venture capitalist, and I'm on the West Coast, and I've done this and this and this in my life, and I'd like you to fly to the West Coast and consult on an investment in a new company. He wouldn't tell me the name of the company or the people involved. That's involved in recombinant DNA. So I even at that stage, I hadn't gotten cook calls. I get cook calls all the time now. But at that stage, I didn't. It wasn't well known. And so I asked him for some references at MIT, and he told me that one of our most distinguished professors in metallurgy had worked with him for decades. And I called him, and he said, anything Ray Schaefer says to you, you can book. So, I'm a pretty gregarious guy. And I like to talk, and I like to learn, and I like to do things that's a little different. So I said, okay. I bought the airline ticket, booked it to San Francisco, and flew out there. And I'm met by Ray Schaefer, and they marched me into a room in which Herb Boyer, Bob Swanson, Idacor, and Riggs walk in. This is the founding group of Genentech, and the first company in '76. And they laid out the synthesis of insulin A and B chain. They laid out the synthesis of growth hormone, and somatostatin, which is a shorter hormone, which they were as a demonstration that you could produce a biologically active human hormone in bacteria. And this is after I had gone to, I went to Asilomar. So, Asilomar was the moratorium in '75. And Dave Baltimore down the hall from me had been one of the instrumental scientists in asking the National Academy to create a moratorium so we could think about recombinant DNA and how to do it safely. So there was a moratorium. No one was going to do experiments for 6 months using recombinant DNA. And the Asilomar conference was then to debate how to do that in a socially conscious way, taking into account safety. And so I called Paul Berg, who I knew from my time on the West Coast, a great friend, and said, can I get into this? And he put my name on the list. So I flew out to Asilomar and was there and listened to all the debate about recombinant DNA. I wanted to do recombinant DNA, and we were putting together a P3 lab. Before P3, we were debating this. So I went out, and they made the investment in Genentech. And then with conversations with Wally Gilbert at Harvard, because Wally was one of the leaders in this whole field of science, with the venture capitalist And we invited a bunch of European scientists, Charles Weissman, one of the fathers of molecular biology, Ken Murray, wonderful guy in recombinant DNA, just a number of other people, and held a scientific conference. This is fun. These are really people that you want to know. And sure, that's, and our motivation was, at least my motivation, Wally's, which we talked a lot about because we flew back and forth across the Atlantic together all the time, was to bring this laboratory academic-based science into the private sector, where we could have people become professional at doing recombinant DNA and translating our science into something useful, because you can't do that in a research lab, even at MIT. So we met with the European scientists, ultimately, and they were very leery of all of this. Didn't trust the venture capitalists, didn't trust the business people. And, but in the second meeting, they decided we would form a company and the scientific advisory board would have a lot of authority. And we did, and we were all on the scientific advisory board. Wally Gilbert was chair. And so we formed a company. It was in Geneva, Switzerland, in '78. And then in early '80, it moved into Cambridge, about 2 blocks from MIT. And I helped recruit people. I became chair of the scientific board. I was on the board of directors for 29 years. You know, I saw the science progress all the way through MS and treatment of multiple diseases, cloning interferon. Long story, I can tell, about Charles Weissman, and it was fabulous. I wouldn't trade it for anything. I got to know those people well. You notice a lot of things about me. I like engaging with people.
Yes. It's very obvious.
I do, and I get a lot of satisfaction. And I can tell you, this COVID last year and a half has been one of the more stressful times in my life. It really takes a lot out of life when you can't, you know, talk to people and be as active as you would like to be.
Absolutely. So let's bringing the conversation full circle back to Kentucky. Going back, your relationship with Kentucky, the community, how they relate to you. I'm sure they appreciate and they celebrate you. So the story about your relationship with Kentucky, that's the last question.
Well, as I told you, my mother's family lived in this region of Kentucky for about 150 years. And she was one of 6, so a very large family. I have 20 or so cousins, and we have a reunion every year. I go back and immerse in the reunion. I'm, you know, up in Boston. They have a very different life style than I do, but I keep in contact with the community. When I got the Nobel Prize, they named a school after me. So in the community, it's a junior high school. It's literally 200 or 300 yards from my uncle's house where he lived. I've been back there, I've given talks there, I give talks at the high school. And then the road I grew up on, they named after me. And then more recently, I started a scholarship program for 2 of the kids, each year 2 kids who graduated from that high school class. It's a scholarship that allows the kids to stay on campus. So I pay for their housing by giving them a 4-year scholarship to live on campus. And it's it's STEM, and it's one male, one female out of the senior class that graduates. And the idea here is to encourage students from that school to go on to college and to have peers who go on and turn part of that community. We've been doing that now for, I think, 8 or 9 years. Every summer I go back and have dinner with those students. I just had dinner with them in August. It's really fun to see those kids grow and finish school, and some go to dental school, some go to medical school. Many are engineers. My classmate runs the foundation that I don't get involved in selection. I don't, but I require the superintendent of the county and the principal and the head of the science departments to select these students. So I want them to talk about the quality of students they're producing. So it's been, I enjoy it. It's a little way of giving back. I do other things as well. But I still have a lot of people back there.
Absolutely. Oh, this has been a wonderful conversation, and again, walking through your life's experiences, a lot to learn from. And thank you for generously sharing your experiences with us. And I'm sure we're all thankful. And let's have a round of applause for Professor Sharp. Thank you very much. Thank you. This concludes the program. Thank you.